Hello, everyone, and welcome back to this week's lecture. This week we'll be talking about the digestive and muscular systems expanded. So we'll be going into a little bit more detail or covering a lot of the smaller details that we may have not covered in these two biological systems, respectively, during their lectures during the regular program. Okay. So again, this is my personal information along with my contact email if you ever want to get a hold of me. Again, I recommend going to our GroupMe. Okay, so learning objectives for today is what is the major role of the liver. Usually the liver we kind of bunch up and include into the GI system just because that's like the closest thing it relates to. Next, what other functions do the small and large intestines perform other than obviously the main one is digestion, which we covered in depth already. And how are the muscles of the digestive system controlled? Actually, the muscles of the digestive system and the neurons of the digestive system are actually very, very complicated, and we'll get into a little bit more detail on how those muscles and that whole pathway works. Okay, and what are the differences between different muscle types, and how do muscles burn ATP or energy, right? How do they use ATP or energy? Okay, onto the content. First, we'll be talking about the digestive system. Starting off with the liver. This is a major organ that we didn't touch upon on lectures so far, but it is a very important part of our bodies. However, the majority of its many functions are not tested heavily on the MCAT. There are two functions that are tested decently amount, like a decent amount, and these are its detoxification function and glucose regulation functions. And we may have covered at least a little bit on its glucose regulation, but we'll be going more in depth with that, and as well its main function, detox. Okay. So detoxification. The liver is crucial in breaking down and eliminating toxins and toxicants from our body. We ingest either through our mouths or by breathing or through many other means, many toxins and toxicants into our body. And some of these we do accidentally, some of these we do like unknowingly and accidentally, others we do on purpose. Some of these include medications, like even this could be over-the-counter medications that we often take, or it could be more controlled substances and drugs and narcotics. Preservatives and other additives that are very often found in our food. Some of them need to be detoxed by our liver, right? And just because they need to be detoxified doesn't necessarily mean that they're like poisonous and they're going to kill you, but it means that our body doesn't like them and it can't break them down, so it needs to kind of eliminate them and treats them as kind of like toxins if they build up and stay there too long. Another very common toxin that we often consume on purpose is alcohol, and another one is caffeine. Okay, and there are many more, but these are just some common examples that you probably know in your everyday life. The first step to detoxing the liver is to neutralize or break down the toxins into more manageable intermediates. For example, alcohol is broken down into acetaldehyde and then further into acetate by certain dehydrogenase enzymes. These are called alcohol dehydrogenase and acetyl dehydrogenase, or ADH and ADLH, I believe it is. And these enzymes could be, like in this case, they could be dehydrogenases, or they could be some other enzymes that basically they just break things down from a very complex form that we can't really work with to something a little bit more manageable. Okay. However, oftentimes these intermediates are also very toxic, and the body must get rid of them quickly, or there'll be a buildup and they'll cause damage to actually your cells in your body. One example that many of you may have seen is acetaldehyde, which is I mentioned is the intermediate when you're breaking down alcohol. It's very toxic in its own, and especially people of East Asian descent who have a poor functioning acetaldehyde dehydrogenase, which is the dehydrogenase which breaks down acetaldehyde into aldehyde. And people from East Asia usually genetically they don't have a very high functioning acetaldehyde dehydrogenase, and so because of that, when they drink alcohol, they begin to feel very strong symptoms of the toxicity of the acetaldehyde because it builds up because the body can't really do too much with it. And this is like the red flush, the nausea, the hot flashes, all of that stuff that you may associate with alcohol, and to a much higher degree because you can't break down the intermediate and you can't get rid of it quickly. Other toxic intermediates may also be free radicals, which can damage DNA and increase risk of cancer. This is the oxygen-free radicals, which you may remember from our DNA damage unit, or if you've learned about anything cancer-related before. Free radicals actually damage the DNA itself, and this heavily increases the risk of cancer. And our body has repair pathways, which we did talk about in our DNA repair lecture, has repair pathways that kind of try and fix these damages and neutralize these free radicals. But by introducing a lot of free radicals into our body through these toxins, that kind of overwhelms the DNA repair pathway, and there's more of a chance that there'll be a mistake, right? And that increases the risk of cancer. And all of this detox, it all happens in the liver. The liver is the main organ. And so that's the reason that for people who use a lot of drugs, who drink a lot, or who have a lot of chemicals or other substances that they put in their body, either through their food or their drink or anything like that, they can be at a high risk for things like cancer, but also different diseases of the liver, because these toxins can damage the cells of the liver. For example, for alcohol, one of the biggest risk factors for liver cirrhosis is alcohol consumption. So because the liver takes care of the detox, it is also the most at-risk organ if somebody does happen to take in a lot of toxins. The main things to note from this, at least first step of detoxification, is that the first step is always in the liver to neutralize and break down the toxins into more manageable intermediates. But once they're in the intermediates, you still want to get rid of them because the intermediates are often pretty toxic themselves as well. And if you ever see something like this on MCAT, they'll tell you there's this substance that in your liver gets broken down into this intermediate or something like that, and you just have to answer questions based on that. They'll give you the example, so you don't really have to memorize like alcohol gets broken down into acetaldehyde and by what, right? What kind of enzyme, what's the name of the dehydrogenase or something like that. Okay, so let's continue on. Once the first step has been completed in the liver and the toxins have been reduced to intermediates, a process known as conjugation, using amino acids or amino acid derivatives such as glycine, glutamine, or taurine, which is not an amino acid, but it is sort of like an amino acid, so like a derivative, converts them into easily eliminable end products, which can be sent to the kidneys or to the bile, which will lead to removal via urine or feces, respectively. Right? The only two ways we can excrete something out of our body, actually there's three ways. It's either back out through your mouth, either through breathing or by vomiting, which usually we try not to do, or through urine or feces. The other two ways, and that's usually what we do. So you break these down into something that's very easily eliminable. So for example, like sometimes carbon dioxide, water, other sugar molecules, whatever. And then you just send them to the kidneys, and then from the kidneys, those chemicals will be kind of washed out and filtered out, and they'll go through the bladder and out the urine. Or you'll send them to the bile, which will lead them to get rid of through the rectum, right, and in the stool. Okay, so fairly simple, and we're not going to go into for the MCAT purposes, it's fairly simple, but what you may have to be aware of is that they may give you a mechanism, like for example, the one I gave you with alcohol, they may give you a mechanism for another chemical or some medication or something like that, and you have to recognize how it's being broken down, and that requires a lot more biochemistry knowledge rather than necessarily knowledge of the liver functions, right? So be on the lookout for that if that ever shows up. Next up is the liver and its glucose regulation. So the liver is a very large organ, and one of its main functions is the storage of glucose as needed. Using the signal molecules like insulin and glucagon, the liver will either take in or release glucose to and from the blood, right? When we talk about sugar, we're usually talking about our blood sugar, and that's usually what you measure. If you have diabetes, or how you know you have diabetes, is the blood glucose level. As you remember, glucose is stored as glycogen in the body. When you're not using it, it's stored as glycogen. And the glycogen is actually stored in the liver. That's the site where glycogenolysis and gluconeogenesis occur, where you take the glucose to the liver, and in the liver it gets turned into glycogen, and then the glycogen, when needed, gets turned back into glucose in the liver and then sent back out to the blood where it needs to go, right? The insulin glucagon receptors on the liver are very crucial to regulating the blood because that's the site of gluconeogenesis and glycogenolysis I mentioned, right? Again, a lot of this, just so you know, it does happen in the liver, and the liver kind of, if it gets damaged, it may, like if the sensors in the liver get damaged, it may very heavily impact the rates of gluconeogenesis and glycogenolysis. They ask you that, something like that. But for this, mainly you do have to remember what is gluconeogenesis, what is glycogenolysis, what are the steps, what are the conditions, things like that, right? But no, again, this happens in the liver gets damaged, and the receptors, especially if the receptors for insulin and glucagon on the liver get damaged, that could lead to heavy problems in glucose management. Either you won't have enough blood sugar, or you'll have too much blood sugar and be diabetic. Okay, now that we've talked about the liver, and again, the liver has many functions, many like more detailed functions, but we're just covering the main ones, okay? All right, moving on to the large intestine. And while we think of our large intestine as just an organ, it's actually a sprawling ecosystem for many different types of bacteria. It's estimated that about 300 to 1,000 different species of bacteria can live in our large intestine. More commonly, there's about 50 different species that make up like 90%. 90% of our bacteria in our large intestine are within 50 species, but there can be as many as 300 to 1,000 different species living in our large intestine. So it's essentially a whole ecosystem down there, right? The vast majority of these bacteria are anaerobes, meaning that they do not need oxygen to make energy. They just need glucose and other molecules. They don't use oxygen, okay? That's different from our cells, which are aerobes, right? These bacteria have a symbiotic or mutually beneficial relationship with us. Remember, symbiotic means it's good for both parties. They greatly help both our digestive and immune systems. And in exchange, they get to live in our stomach, and they have an ecosystem there, and they sometimes eat scraps of our food, okay? So what are the benefits of bacteria? Usually when we think of bacteria, we usually think of bad things, and we don't want any bacteria in our bodies. That's how we get sick. But in your mouth, in your large intestine, everywhere, you have a lot of bacteria, and a lot of these bacteria are actually very good for you, right? Firstly, these bacteria greatly help the immune system and overall protection of our body from pathogens. Pathogens are like bacteria and viruses and things like that that will cause diseases in us. These bacteria that live in our bodies are not pathogens. They don't cause disease. They just live within us. And because of that, they cover nearly the entirety of our habitable large intestine. And thus, they kind of outcompete any foreign bacteria that we could ingest, right? If, let's say, we have our large intestine like here, and all of this is taken up by our good bacteria, which live in our intestine, any foreign, do it in red, any foreign bacteria that come in will have to take up one of these spots. These spots essentially are all already taken by our good bacteria, and our good bacteria will kind of like not let them stick on us, and essentially they won't let them take hold. And once they take hold, they'll start multiplying and they'll start doing whatever they want, and that's how we get sick. But our bacteria kind of take up all the space, and they prevent that from happening. In addition, our gut bacteria may also release cytokines, just like our immune system does. Like our killer cells also release cytokines to kill bacteria and viruses when they see them. Gut bacteria also release cytokines. Some gut bacteria release cytokines to actively combat and kill the foreign pathogens, because obviously it's in their best interest not to lose the fight to these pathogens either, because if they lose, they will give up their spot, and the bacterial and the foreign invaders will take over. So they want to keep their ecosystem to themselves, and so they will protect it, right? And that's good for us too, because we also don't want these foreign invaders living in our stomachs. Another great benefit of the gut flora is their aid in our digestion. There are certain polysaccharides, starches, sugars that we eat that our bodies can't break down. But the enzymes provided by these gut bacteria can break them down. And once they break them down, we can actually use them and get energy from them as well, right? Without these bacteria ingesting these molecules, would lead to low benefit. Without these bacteria ingesting these molecules would lead to low benefit for us. Thus, we would need to consume a lot more food in order to receive the same caloric benefit out of it, right? If you eat a bunch of like wood off like a tree, you can eat and eat and eat and eat, and you won't really get much benefit out of it because you can't digest the wood, right? And just like you can't digest like rocks or something like that. It'll go in and go right out. Same thing with these polysaccharides and these sugars that our bodies normally can't digest. If you couldn't digest them and you just ate them, they'd go in and then they'd just go right back out, and you wouldn't get any benefit from it. But because these gut bacteria live within us, they kind of break them down a little bit, and then we kind of take a little bit for ourselves, and we can use something out of something that we couldn't have before. There have been studies that shown if you completely get rid of these gut bacteria in other mammals, like in rats or whatever, that they will have to eat many, many more calories per day in order to keep the same weight, in order to prevent themselves from losing weight than the same exact mice or rats if they just had these bacteria. Okay. Going into a little bit of medicine, these benefits are one of the main reasons why it's advised for medical professionals to not overprescribe antibiotics. So antibiotics kill bacteria, and it may seem like, oh, what's the harm if you're just killing bacteria? Well, firstly, one of the main reasons that they say not to do that is because the bacteria will grow, as a species, will grow resistant to the antibiotics, and over time, our antibiotics will stop working. But another big reason is that you actually need bacteria in your stomach to, like, you can see all of these benefits. You actually need these bacteria; they're actually helpful for you. So if you just keep constantly taking antibiotics, the antibiotics don't discriminate between bacteria, so they'll kill everything. And if you take in too many of those when you don't need it and it's unnecessary, they'll kind of eliminate many of the gut bacteria as well. Similarly, this is why it is also healthy to consume probiotics, right? There's antibiotics and there's probiotics. Probiotics are things like yogurt and other like dairy products that have, or sometimes now we have like just special like probiotic things which have just a mesh of bacteria in them. But natural probiotics are like yogurt and stuff. And these probiotics contain a lot of these healthy bacteria, especially now with more modern science. We can actually control and determine which exactly bacteria species we want in there. So these probiotics replenish your gut flora and make sure you have a healthy amount of those, right? Next, let's talk about the muscle control of the GI tract. So our GI tract has a very complex system of neurons controlling it called the enteric nervous system, or ENS. Many consider this kind of like a second brain, as it's independent of the brain itself and the spinal cord. Although it can be influenced by them via the parasympathetic and sympathetic nervous system, parasympathetic nervous system is more like the rest and digest. This can influence the stomach to, okay, start digesting stuff now. We're resting, we digest. Where the sympathetic nervous system can tell them this is not the time for digesting. We need to fight or flight or something like that. So they can be influenced by the actual main nervous system, but even if you cut off all, like, and there have been experiments where on animals they've completely cut out the connection between the main nervous system and the enteric nervous system. And the enteric nervous system still continues to function, although at a reduced capacity, of course, because again, you do need the parasympathetic and sympathetic nervous system, but still it continues to function even without any outside interference at all. That's why people call it like a second brain. One of the main jobs of the ENS is to control peristalsis, which is the muscular contraction of the GI tract used to move food along it, right? So you need to move food from like your mouth down to your stomach, and you need to move it past. So how do you do that? Well, you just take the ball of food, and then it's going down the tube, which is your esophagus, and then your GI tract. Essentially, you'll just squeeze it, then squeeze here, and squeeze here, squeeze here, then keep going, keep going, keep going. And the food, when you squeeze it, it'll move to the right, to the right, to the right, to the right. It'll essentially like, kind of like when you squeeze like toothpaste out of a tube, it'll go right out along your GI tract, right? And this squeezing is called peristalsis. And peristalsis is essentially completely controlled by the ENS. And what that means is, you know how when you have your brain going to your skeletal muscle, it starts in your brain, then it goes down the long axon to the end plate, to the axon end, and then there's the acetylcholine going to the muscle, and then it excites the muscle, and the muscle begins contraction. Well, this all happens exactly the same way, except it doesn't start at the brain, it starts in this complex web of neurons called the ENS. So it starts in your stomach, and then the stomach releases the shock down the axons or wherever, the equivalent of the axons, to the GI tract muscles. Again, go to the motor end plate. Acetylcholine goes to the muscles contract. Peristalsis occurs. Okay, so peristalsis can happen completely independently of the main nervous system. Without peristalsis, the food bola, which is like a ball of food, right? When you chew it and swallow it, it turns into a ball, like a mush, and it's called a bola. Without peristalsis, the food bola would never move through the esophagus or the intestines, and the full muscular control process is regulated solely by the ENS. Okay. All right. So the ENS is also crucial for controlling the secretion of necessary hormones such as secretin, which signals for the release of pancreatic and stomach enzymes needed for digestion. We talked a lot about those in the main digestive system lecture. All of those are released by secretin, and what determines whether or not secretin is released is ENS, right? Next up, let's talk about the muscular system, okay? So first, let's talk about peripheral circulatory assistance. As you know, we have a vast network of veins and arteries that carry blood throughout the body. The veins and arteries that exist outside the chest and abdomen, so for example, in the arms, legs, etc., basically your appendages, right, outside of this like torso region. And this is called the peripheral vascular system, or the PVS. As these are fairly far from the heart, they require some assistance from the muscular system to efficiently pump that blood back, right? The heart is really strong pump, really strong muscle. It just pumps, and the blood goes from the heart, and then it goes out. But as it goes out, it also obviously loses power, right? As it goes farther and farther out, it loses more and more power. And especially when you're talking about the veins, which, remember, arteries away from the heart, veins going back to the heart. The veins have already lost most of the power, especially in this peripheral system, which is far from the chest and abdomen. So you need a little bit of help to kind of have enough force to send it right back to the heart without any problems. The contraction of muscles in your arms and appendages, whatever, is crucial for this movement of blood through the venous system back to the heart, right? Next, and again, this is to prevent like things like pooling or whatever else in your blood, right? When you don't move your body for a long time. Next, let's talk about thermoregulation using our muscles. So normally when muscles contract, they always release some of the energy consumed as heat. Normally, this is considered a waste, as we want this energy doing work for our muscles and not just leaving in the form of heat. if you remember from, or if you learned from thermodynamics, energy, not all in a machine, not all of the energy released by the machine will be doing work, right, as intended. So machines have like a certain efficiency, how much of the energy from this machine or from the system actually does work. For our bodies and for our muscles, it's also like a machine. It's not 100%. And according to physics, no machine can be 100% efficient where all the energy goes directly to work. Our bodies are no exception. When our muscles release energy, or when our muscles like do anything, some of the energy that's supposed to be doing work is actually lost in the form of heat. Usually that's kind of like the waste product, and we want to reduce that as much as possible. However, there is a situation in which actually we try and use this, and we actually want to increase this percentage of heat release, and we want to release as much heat as possible, and that's when we're trying to warm up our body in response to cold. And what we do is we undergo a process called shivering, which I'm sure many of you have heard before. When shivering, the muscles begin to repeatedly contract and relax with the goal of releasing as much energy in the form of heat as possible. Now, this waste product of heat is actually now the main product of the goal, right? When you move your muscles really, really fast, a lot, you'll release, your muscles will be moving, so you'll be doing a lot of work, but you'll also be releasing a lot of this heat as a byproduct. And that's heat is what we're exactly looking for, and that heat will be released into our bodies and thus warm us up. The shivering process is an automatic response to low body temperature, meaning it's kind of like an instinct, and it's controlled by the automatic nervous system, which is present mostly in older children and adults. The reason that's important is that infants and young children have not developed this response yet, and so they actually have a large supply of brown fat, which is different from the white fat that we have. Mostly the white fat that we have very low brown fat, but young infants and children have a larger amount of brown fat, and the brown fat, when it burns the energy, like normal fat, when you use it up and burn it, it's mostly used up it's mostly all used up, whereas brown fat releases a lot of heat when it's burned. It's not very efficient, but again, in this case, that's what we're looking for. And the elderly begin to lose this response with age. So for them, they actually don't have brown fat, so they actually have like struggle a lot more with the cold, right? As you get older, you struggle a lot more with the cold and you need a warmer environment to live in, because you can't regulate your body temperature that well anymore, because you lose, for example, the shivering response, among other things. The response is initiated when we are faced with external cold, like in the winter, obviously, but it's also present during illness, and that's what we call chills or rigors or something like that. Because during illness, our body's goal temperature increases. So, for example, the normal temperature is, our internal body temperature is 98.6. Our body right now, for me, I'm not sick. My body says, okay, I want a goal of 98.6. Now it checks, what's my body temperature right now? Okay, it's 96, right? So it's like, I'm a little bit under the goal. So that means I need to heat up a little bit to get to 98.6. So it activates all of these automatic responses, include one of which might be shivering, and that releases more heat, and my body goes up to the 98.6 that it's set as the goal. And once it does that, it's like, okay, we're good. I just have to maintain it, no, nothing is required. Now imagine I immediately right now just got sick and developed a fever and had a bunch of pyrogens released in my system, and I became like sick with a fever and chills. What would happen is my actual temperature right now doesn't change, right? When you have a fever, your actual body temperature doesn't change. What changes is actually the goal. Instead of this 98.6, your body actually raises the goal temperature to, like, say 105, right? It says now my new goal is 105, and then it looks again. Okay, the goal is 105, but right now I have 98. So that means I must be kind of cold. I need to warm up, right? And so it now actually again triggers this shivering response among other responses to warm you up and get you up to 105, right? Because that's the new goal. And that's why when you have a fever, even though you're actually really hot, because you're probably at like 100, which is higher than you normally should be, but it's still under what your body wants you to be, which is like 105. So that's why you still feel really cold, because your body's telling you you're underneath the goal body temperature. You're cold. You need to warm up. Even though you're actually physically warmer than you should be. Okay. And again, that's just kind of explained in this, right? Okay, now let's talk about the main thing of muscles, which is fiber types. Our muscles are made up of, it's not just like one long muscle, it's not like one string and that's your whole bicep. Our muscles are made up of a network of fibers that all work together. These fibers come in 3 variations, and different muscles have different ratios, compositions of these fibers, right? The percent of each fiber in each muscle is going to be completely different. The types are type 1, which is called slow oxidative, has weak contractions, it's very resistant to fatigue, meaning you have a lot of stamina and it doesn't get tired very easily, has many mitochondria in it, and the fibers are very small usually. And the oxidative refers to that it uses oxygen and it uses oxidative means of getting more energy. There's type 2A, which is fast oxidative. Again, this uses oxidative methods of getting energy, and so again, it requires oxygen. It has high contractions or stronger contractions, faster contractions. That's what the slow and fast mean. So stronger and faster contractions, but it fatigues more quickly, has less stamina. It has a medium amount of mitochondria, so not as many as the type 1, but still some mitochondria, and the fibers are much larger than the type 1 fibers, like physically larger. And then there's finally type 2B, which is fast glycolytic. This glycolytic refers to the fact that it does not use oxygen, does not use any oxidative means to get more energy. It just uses glucose, right? Only glucose and nothing else. This has the strongest and fastest contractions out of all, but it also fatigues the quickest. It runs out of energy the quickest. And that's because getting energy just straight off of glucose is very difficult, and you won't have a lot of energy if you just use glucose, but it's very fast. You can get a lot of energy very quickly from glucose, but in the long term, you're not going to get that much energy from glucose compared to oxidative means. Because it also doesn't use any oxidative means, like, for example, doesn't undergo the Krebs cycle or ATP synthase or anything like that, or the electron transport chain, doesn't need that many mitochondria, so has the least amount of mitochondria. And again, the fibers themselves are physically really large. The fiber composition of different muscles in our body varies greatly. So the composition of your bicep, your pectoral, and your eyelids are very different. But the fiber composition in the same muscle between different people also varies greatly based on genetics and training. For example, the bicep muscle in me and somebody like a professional athlete would vary very greatly in terms of what we have. And even among athletes, varies very greatly based on both what type of training they do, so what kind of sport they play, what kind of sport they're trained for, as well as just what are their genetics. Okay, so type 1 fibers are found predominantly in muscles that are always active, like for example, the diaphragm and the muscles of endurance athletes. For example, marathon runners, long distance swimmers, long distance cyclers, etc. These are muscles that you don't really need that much like explosive, like fast, strong contraction, but you need to be contracting very, very, very for a very, very long time. The diaphragm, as well as like the legs, arms, body of like runners, long distance runners and swimmers, needs to be active for a very, very long period of time. Compare that to type 2 fibers, which predominantly compose muscles that need fast and accurate and strong contractions, like for example, your eyes. Your eyes need to be able to move very fast to track things. Your eyes need to be able to see everything. They need to contract very fast. So your eyes use predominantly type 2 fibers. Like I think 80 to 90% is type 2 fibers, as well as the arms and legs and other muscles of power athletes. So power lifters, sprinters, etc. People who have explosive and like very strong and fast contractions for a very short period of time, but they don't need it for a very long period of time. So for example, power lifters who only are lifting one thing for like maybe 5 seconds, but they need to lift a lot. Or sprinters who need to run for like a minute, but they need to run very, very fast for that 1 minute. And you can actually see this actually physically. Remember that type 1 fibers are much smaller than type 2 fibers. So here you have comparison of a men's marathon runner and a men's sprinter. And if you want to just look at their leg muscles, both of them are very physically fit, obviously, because they do this sport professionally. But if you just look at the pictures, this guy's legs look much more muscular and much stronger than this man on the left, the marathon runner. But the marathon runner's muscles are actually also very, very well defined and very strong, but they're strong for a different way. So they're much smaller because the majority of his legs is type 1 fibers, because he needs mostly type 1 fibers in order to be able to run 26 miles very fast, right? He needs to not burn out after like the first minute. So he needs to have a lot of type 1 fibers, which are typically tend to be smaller. And so his legs look a lot smaller in terms of size compared to the sprinter, whose muscles are much larger, but that's because he actually has mostly type 2 fibers, which are useful obviously for sprinting for his sport, right? So they both have a high amount of muscle and very well-defined muscles, but for just different sports, for different activities, different compositions, because they need different functions essentially. Okay. All right, so muscle energy sources. As mentioned, the energy sources from muscles come from either oxygen or solely glucose. Oxygen is the most efficient way to create energy, as one run of glycolysis Krebs cycle will produce much more ATP per glucose molecule than simply glycolysis, which we talked about many times before. For this, you need an abundance of oxygen coming into the cells. The oxygen can only come in from the lungs and must be transported to the muscles by the blood. There's no other way to get oxygen into your muscles because you can't like breathe in through your skin. At least humans can. Some animals can't. Thus, when undergoing heavy exercise, breathing rate and heart rate increase dramatically so that you can get more oxygen from the air into your lungs and then from your lungs to your muscles very quickly. Without it, the muscles will fatigue and fail to contract, as ATP is required for muscle contraction, and ATP is mostly made from oxidative purposes, such as the Krebs cycle. And again, for this, an abundance of mitochondria is also required, as it so that more oxygen glucose can be used simultaneously without a bottleneck. Imagine if you had 100 molecules of glucose with all of the oxygen and everything you need to run through all 100 Krebs cycles at once, but you only had one mitochondria, and you know Krebs cycle runs the mitochondria, so the mitochondria takes in one glucose and oxygen at a time and runs through them. It's going to be very slow. It's going to be a huge bottleneck. So you need a lot of mitochondria so you can run a lot of Krebs cycles at the same time. Okay, makes sense. All right, that's it for today's lecture. Hopefully it wasn't anything too complicated. It's mostly stuff that we've covered before, but just a little bit more detail and kind of supplement, as you will, with the name of this series, to what we've learned before. Hopefully this helped you learn something new. And these aren't very common on the MCAT, but they do appear sometimes, so it's great to know. All right, if you have any questions, again, please feel free to email me. Set up office hours, or ask on the group me. All right. Thank you very much for your time. Have a great night.